Hi, I'm Krista Watson, and this is my mini exam video 6. Today I will be addressing prompt 2. We will be examining the developmental network of the regulation of skin spikes. So first we're going to look at the wild type of this developmental network. The S protein promotes the P protein, while the P protein inhibits the I protein, so that the I protein cannot inhibit the S. So E promotes spikes, and the wild type will have spikes. So now we're going to look at what happens when the P protein is absent from the sequence. The P cannot inhibit the I, so therefore K is blocked by I, and K cannot activate the E to produce the spikes. So this will have no cells that have spikes. Now we will be looking at what happens when I is lacking from the developmental network. Since I inhibits K, K is able to be expressed, and so E can be expressed, so spikes can be expressed in transcription. Now we will be looking at what will happen when I and E is lacking from the developmental network. Since I is upstream from E, its effects are not as important as E's. When E is lacking, it cannot promote the expression of spikes, so this cell will not have spikes. Now we'll be looking at the effects of S and K lacking from the developmental network. Like the example before, S is upstream from K, so its effects are not as influential as K lacking from the developmental network. But since K is lacking from this developmental network, K cannot activate E so that spikes are produced, so this skin cell will not have spikes. So lastly, we'll examine which different forms of regulation can affect which cells in the body have the spikes present or not. A developmental network is an example of a sequential gene expression where the expression of one gene can affect another in a series of events. And then one thing that can affect which cell, where some cells in the body will have an expression of a gene and others won't, is regulatory proteins interacting with an operon. This can control the expression of transcription, and the concentration of these proteins can vary throughout the body, which could lead some cells to be inactivated or activated through regulator proteins.